Genetics and bioengineering are fields that have recently been the subject of many controversies, particularly in their relationship to medicine. In order to explore these issues, however, we must first define some terms. What do we mean when we talk about genetics or bioengineering? A gene is a specific sequence of nucleotides in DNA or RNA that is the functional unit of inheritance, controlling the transmission and expression of one or more traits by specifying the structure of a particular polypeptide or controlling the function of other genetic material. This ties into genetics, which is the study of genes, heredity, and variation in living organisms. Bioengineering refers to the biological or medical application of engineering principles or engineering equipment. Bioengineering includes a wide variety of subfields. These include biomaterials, biomechanics, cell and tissue engineering, biomedical devices, biomedical imaging, computational bioengineering, and synthetic biology. To summarize, in the context of medicine, genes are hereditary contributors to our health. Genetics is the study of these genes, and bioengineering is the application of biology, including genetics, to solving real-world problems. In the US, one of the largest controversies surrounding the fields of genetics and bioengineering is patented genes. Patents are granted by the US government to inventors for new and useful inventions and discoveries. A patent grants to its owner the right to exclude others from making, using, or selling a machine or composition of matter, or using a patented method, typically for a period of 20 years. Patents require disclosure that teaches the world how to make and use an invention. In return, they reward the inventor with a period of time during which profits may be earned from its commercialization. Nearly 30,000 human genes have been patented in the US. The first type of genetic invention covers testing of genetic differences. Since the fundamental discovery was the observation of a genetic difference linked to a physical difference, such as the occurrence of disease, then any method for testing for that genetic difference can be covered by the patent. Well-known examples of disease gene patents include those covering genes identified with breast and ovarian cancers, colon cancers, cystic fibrosis, and hemochromatosis. One of the primary concerns about human gene patents is that they will make it more difficult to perform research, thereby delaying or impeding discovery and development of diagnostics and therapeutics. However, it is still uncertain whether gene patents are beneficial or detrimental to scientific research and commercial product development. Another controversy that has attracted media attention is stem cell research. Stem cells are cells from which all other cells with specialized functions are generated. Under the right conditions in the body or a laboratory, stem cells can divide. These daughter cells can either become new stem cells or specialized cells such as blood cells or brain cells. No other cell in the body has the natural ability to generate new cell types. Studying stem cells can help in understanding how diseases occur and open the possibility of generating new cells to replace damaged or diseased cells. They can also be used in drug testing, especially after being programmed for cell types that are not otherwise obtainable. Stem cells are divided into two main types, with a third discovered only recently. Embryonic stem cells are found in embryos that are 3 to 5 days old. They are the most versatile type of stem cell. Embryonic stem cells come from donated eggs and are fertilized at in vitro fertilization clinics. Contrary to popular myth, embryonic stem cells do not come from aborted fetuses. Adult stem cells are found in small numbers, usually in bone marrow or fat. Their ability to give rise to other cell types in the body is much more limited than embryonic stem cells. For example, stem cells found in bone marrow usually only give rise to blood cells. However, recent studies suggest that these stem cells may be more versatile than they appear. Further research is necessary. A very recent discovery found stem cells present in the amniotic fluid of pregnant women. But currently, research is very limited and the exact functions of these cells remain unknown. Most controversy regarding stem cells comes from the use of embryonic stem cells for research. the extraction of stem cells usually destroys the human embryo, leading to some ethical questions regarding embryonic stem cell research. Genetics and bioengineering are rapidly developing fields. However, they are surrounded by many ethical questions that should be taken into consideration.